Now we're covering active transport. This has to do with when a cell brings particles or fluids across the cell membrane, form vesicles out of these, and bring them inside the cell. This is called endocytosis. Let me get my little pointer. This term, pinocytosis, which means cell drinking. Anytime a cell brings a substance across that requires some fluid along with it, that's what it's referred to. This is a really good diagram. So it says pinocytosis, but here is the invagination of the cell membrane. Here's the extracellular fluid, the ECF. That means the fluid outside of the cells. And then here's that little structure inside. So when a cell brings that across, it's got fluid in it. It's called cell drinking or pinocytosis. Phagocytosis, which I'm going to bounce back just for a second, which is what this is previously. That's bringing a large particle across like this. And same thing. Here's the invagination. And then you get a double layered vesicle. In this case, it says vacuole, just a little bit larger than a vesicle inside the cell. Receptor-mediated endocytosis is a very specific process. Here you have receptors. See, the blue structures will not fit here. These star-like structures fit perfectly. So they come down here, they bind, and then the cell brings them across in this vesicle that's very specific. That's driven by the receptors. So unique receptors in a protein coat that fold inward and pinch off to form a vesicle. Now, bear with me again. Just to give you a little better, bigger font, but here is, phagocytosis, pinocytosis, receptor-mediated endocytosis. This one, very specific. These two, non-specific. Just bringing something across from an invagination of the plasma or cell membrane. This one's called cell eating, this is cell drinking, and this is a very specific process.